And lovely, Kate, would you like to introduce Lucy for us?
Yes, so it's my pleasure to introduce Lucy Davidson, who is a MRC fellow at the Royal Veterinary College in London, and is also part of the Department of Physiology, Anatomy and Genetics in Oxford. I won't read her whole bio because that's available if you want to see it, but she's been working on canine insulinomas, and I think this is a really interesting area that, as scientists working in the NET field, we haven't really sort of communicated with quite so much. And I heard a few talks recently and I find it really fascinating. So I'm really looking forward to hearing her talk today. So Lucy, please go ahead, and thank you for agreeing to give this talk.
Thank you very much indeed. Can everyone hear me okay? Yeah, good. Okay. So yes, my name is Lucy Davidson. I'm actually a vet. I see dogs and cats as my patients. I'm a specialist in internal medicine. A lot of my patients have diabetes and other endocrine disorders, but I'm really keen to develop what we would call a One Health approach in endocrine and neuroendocrine disorders, because I do think there's a huge number of really valuable, interesting, and potentially relevant models that we see in our companion animal species. As I'm sure you're all aware, there are many people these days who want to work up their pets to a very high level. They do want to explore treatments, but we do also have the opportunity as well to bring people together to understand how breed-specific conditions work, and sometimes that also gives us a bit of an insight into the genetics if we're only seeing things in certain breeds. So I'm just going to try and give you a flavour today. What I've also done is I've put within this talk some key papers that I think if anyone is super keen to go and find out more about canine insulinoma, they would be the ones I would start with. But I'll also put my email address at the end, so if anybody wants to reach out and have a conversation with me or Florent Bouchand, who's one of our surgeons at the RVC who has a special interest in insulinoma and other endocrine tumours, then please do let me know. Before we start, I just want to acknowledge a few places, a few people. As you've already heard, I work at the Royal Veterinary College in London, which you can see here on the left-hand side. Historically, I've also been at the Wellcome Centre for Human Genetics in Oxford, currently at DPAG in Oxford, but actually some of the patients you hear about today are also from my time working as a clinician at the vet school in Cambridge. You never really leave anywhere you work, so we stay connected because some of these patients are really quite rare. Very briefly, just want to give you a little clinical flavour of the type of things that I see in internal medicine practice. This is an example of a 7-year-old female neutered boxer called Kitty, and she came to us as a referral, second opinion from primary care practice because she was collapsing at exercise. Her owner's complaint was that she got really tired, didn't want to go on long walks, would lie down, but actually sometimes would appear to sort of get very faint. She was also hungry and she'd gained weight recently. And I'm not going to go through our entire clinical diagnostic approach. We try and encourage the students to go through all the different problems and come up with the differential diagnoses for each of these. But if we broadly look at what's going on with Kitty, one of the things we have to really try to do in veterinary medicine is think around the edges with our clinical history taking, because we can't ask the patient how they feel. So we rely very much on owners picking up things that are different about their pets, and also our clinical examination, trying to decide if there's anything there that would point us in one direction or another. So in these sorts of cases, obviously we're thinking about neurological diseases and pain and musculoskeletal diseases, sometimes anemia. We'll see dogs get very tired at exercise. Boxers, if any of you know anyone who's owned a boxer or who have owned one yourself, will realise they also have quite a lot of cardiac diseases. Hypoglycemia is on our list, as is hypocalcemia for exercise intolerance. Boxers also have their fair share of hypothyroidism, so we're thinking about that with the weight gain. They're also brachycephalic, they have squashed face, so we worry about brachycephalic obstructive airway syndrome. And sometimes owners don't really take the signals from their dogs about heat exhaustion in hot weather. Sometimes collapse can be to do with that because dogs can only really sweat through their foot pads. They're not very good at sweating in other ways apart from losing heat from their tongue and their feet. So when we see these sorts of patients, the type of test we will do typically, depending on what the owner wants, we usually will do an ECG, particularly for a boxer. We might even do a 24-hour ECG. We typically do hematology, biochemistry screens, just looking for things like anemia or renal or liver problems. And we will check blood glucose on a fresh sample, ideally when the patient's collapsed or at least a while after they've eaten. We probably will do endocrine testing. And then we rely a reasonable amount on diagnostic imaging to rule out other conditions, but I'm sure many of you are very well aware neuroendocrine tumours don't always show up particularly well on plain diagnostic imaging. So with Kitty, actually, what we saw, no evidence of heart disease. She wasn't hypothyroid. Her blood count was okay. Calcium levels were okay. She didn't have musculoskeletal disease. She didn't have neurological disease. Boxers, unfortunately, do get their fair share of brain tumours and other neurological disorders, but she didn't seem to have anything that would point to that clinically. Her respiratory function was okay. But what we did see on her blood glucose at rest was a blood glucose of 3.3 millimoles per litre. Now that's actually still within our reference range. It's at the lower end of reference ranges. Now, if you're looking at a cat with a blood glucose like that, you'd be really quite worried because cats tend to have a very high blood glucose when they're stressed. They get really bad stress-induced hyperglycemia. Dogs, not so much, but still that's a little bit unusual for us to see a blood glucose at that level. There's a whole literature around what we should be using to measure blood glucose in dogs, because it depends on which glucometer you use as how accurate that is. But actually this was a specific veterinary one. And this is where the question arose, could this be an insulin-producing pancreatic beta cell tumour, so-called insulinoma? And here, in the case of diagnosing insulinoma in veterinary medicine, I think it is likely to be very similar in human medicine, so-called Whipple's triad. We want to get various different elements of the medical diagnosis to add up before we can make that call. We need to know in our clinical patient that there's evidence of chronic hypoglycemia, and that would be usually through documenting a reduced glycated albumin or fructosamine concentration. We want to make sure that the collapse is associated with hypoglycemia, that it's not a seizure or it's not some other sort of cardiac electrical abnormality, for example. Importantly, it also helps us to know if feeding the dog reverses the clinical signs, and then we would make the confirmation diagnosis by an inappropriately high insulin concentration measured when the dog is hypoglycemic. And there are some challenges around measuring canine insulin. You've got to be careful which lab you use. I've been involved in some quality control studies and sending the same sample to about 50 different labs, and it's remarkable how different those insulin concentrations are in the same sample. So that's a sort of warning for anybody veterinary who might be listening to this talk. Diagnostic imaging can help us, but we don't rule insulinoma out based on diagnostic imaging, because they are definitely not always visible as a discrete nodule. Even if the owner wants to pay for CT or MRI, we still don't always see that. Ultrasound sometimes will give us an appearance of a mass, but then surgically it might not look that's the case. And we have to also be aware that there are other things that can cause hypoglycemia in dogs. Some of you may be aware, we're coming up to Easter, where we're all told not to give dogs chocolate because it's toxic. Another thing that's really toxic and can actually mimic neuroendocrine insulinoma in dogs is xylitol toxicity. So some of these sweeteners can actually cause a really dramatic hypoglycemia in dogs. We have owners who have been giving sugar-free mints and things to patients, to their pets, and that might be something that causes intermittent collapse without the owner making that association. Other tumours obviously can cause the hypoglycemia, and there are many other conditions, endocrine diseases, liver diseases. Certain toy breeds aren't very good at maintaining blood glucose even in normal circumstances. And then there are the more rare conditions, or conditions that might come with other clinical signs that would point us away from a neuroendocrine disease. There are, very sadly, case reports in the literature of people inducing clinical signs like this with things like insulin in dogs. We see even in veterinary medicine, we do see Munchausen by proxy type of syndromes, and there is one that's been written up in a dog with hypoglycemia that was thought to have an insulinoma, but it turned out the owner was giving insulin inappropriately. In Kitty's case, our glycated albumin, our fructosamine, was in the low part or below the reference range, so we knew she had chronic hypoglycemia. And again, in something we do in veterinary medicine, not really done in human medicine, we did a 15-minute ball game under close observation, and we managed to recapitulate those clinical signs of the dog becoming much more quiet, much more lethargic, and actually we stopped before there was any collapse. But when that was the case, we could see that blood glucose really was pretty low. It was below the reference range now, gone down below 3.3 to 2.5, and the insulin concentration in that exact same sample was 44 microunits per ml, which is above our reference range, and that's certainly inappropriate for a hypoglycemic dog. So canine insulinoma, I'm sure you're very familiar with human insulinoma. Canine insulinoma is relatively rare. I probably am aware, coming through the vet school at the RVC, of maybe 5 or 6 cases a year. But there are others in various different practices around us, so we are able to gather a reasonable cohort in the space of 12 to 24 months if we want to do a study. Surgical treatment alone is rarely curative, probably because we don't see these presenting till quite late, like a lot of veterinary conditions that are not overt, and there's no verbal report from a patient saying that they feel a certain way. It often is quite advanced by the time we actually are seeing a referral to make a diagnosis. And of course, insulinoma is uncontrolled proliferation of pancreatic beta cells. And I became interested in this as an endocrinologist because actually the far more common beta cell disease that we see is diabetes mellitus, and that is associated with destruction of pancreatic beta cells. And there are two sides of the same picture, really, and we do believe that beta cell renewal and biology and understanding what turns beta cells into a destructive pathway versus a sort of immortalized pathway is going to be important in understanding both diseases. Now, there is a review that was written by one of my colleagues at the RVC about diagnosis, treatment, prognosis of canine insulinoma. It was published last year, and I've put it here. What I also want to point out, that case that I just presented to you was a collapse at exercise, but that's not really the most common presentation. Actually, quite often, it takes a long time before the owners notice that there's a clinical sign of insulinoma, and sometimes these dogs come into us through neurology with seizures. So 50% of dogs in this 320 dog series actually presented with seizures rather than the weakness or the collapse at exercise. And you'll notice here polyphagia, so an excessive hunger and often weight gain is also something that's seen, but it's by no means one of the most common signs. So even in veterinary medicine, there is still a diagnostic challenge. It's, by the time patients come to us, we're alert, we're looking for this disease. In primary care practice, it's much harder to pick up because a seizuring dog, most people would imagine, has epilepsy. We have just completed a study using the Royal Veterinary College Vet Compass portal, which is an amazing resource really for us to look at primary care records. And we have been able to look at dogs who have had a diagnosis of insulinoma or a suspected insulinoma in practice, and that's going to be submitted for publication very soon, so I'm not going to go too much into the findings today. But just as a reminder, the reason that we see this major problem in these dogs is that we have insulin release which is inappropriate. Normally, it's hyperglycemia that causes insulin release, and when you have hypoglycemia, you should have glucagon release to counteract the insulin, and the insulin should switch off. And in these dogs, we have excessive, uncontrolled, autonomous insulin release. It actually, it means that these dogs do store glycogen, and they do the liver does get large sometimes in these dogs and they can put on weight, but they have a persistent hypoglycemia, which is why they're so likely to collapse and why we do sometimes see seizures in these patients. Now, how relevant is this to human medicine? Well, again, I would refer you to this article that was written a year or so ago, a couple of years ago now, in Translational Oncology. It talks about canine insulinoma as a model for human malignant insulinoma. And what it shows here on the right-hand side, I've lifted this figure from that article. It shows the age incidence, not in exactly years matched human for dog, but at least as different parts of the dog's lifespan. So that the dog age range here is 0 to 16 years. The human is 0 to 91. And you can see here that typically insulinoma in dogs and humans occurs in late middle age and older. There are a small proportion of human insulinomas that happen younger. My understanding is, I'm not a human medic, is that some of these younger presenting insulinomas are related to multiple endocrine neoplasia syndromes. Now, we do sometimes see more than one neoplastic endocrine disorder in dogs, but we don't yet have any evidence that that is a specific heritable mutation. So a couple of other things that compare the, before I talk about our research, that compare the oncology of human insulinoma and canine insulinoma. The first thing that they share is a stem cell marker called CD90, which has been identified as a potential therapeutic target in humans. And we know that that's now present in clinical insulinoma samples from dogs. So there is a move towards anti-CD90 monoclonal antibody as a promising treatment for insulinoma that do carry this marker. And you can see this paper was written by Florin, who when she was at Utrecht, but now she's with us at the RVC. So this is an area, I think, of future promise. Similarly, another population of cancer stem cells within the insulinoma is characterized by the expression of a panel of different genes and overexpression of Notch signaling related genes. So that also points us, both in humans and dogs, to a potential targeting therapy towards Notch pathways. And that's certainly something I think that will be valuable to explore. And it may be that in these treatments coming forward, the canine species could provide an opportunity for us to try some of these medications, as long as the owners provide informed consent and we get the relevant ethical approvals. The third thing is the RNA sequencing of canine insulinoma that was done by a group in Edinburgh. And this was just bulk RNA sequencing, looking at normal pancreas and early insulinoma versus late insulinoma. And what they discovered there was that actually the late insulinoma primary tumor looked very, very similar to the metastatic lymph nodes. And the metastatic tumors were predictable by sequencing or RNA sequencing the primary tumor. So we can get some indication from the markers that are expressed by the primary tumor how likely this is to metastasize. So returning to canine insulinoma and that mirror image really of that, which is diabetes. Why do we think those two might be useful to study side by side? Well, this actually goes all the way back to many years ago when I did my PhD in canine diabetes. And I did some work to understand which breeds were most susceptible to diabetes. And we did this by looking at an insurance database and seeing how many dogs of each breed were on that database, and then how many dogs from each breed were claiming on their insurance policy for insulin. So we had an indication of how popular each breed was and how many dogs within the breed were diabetic. And actually what you see here is really, really striking. The odds ratio for diabetes, if you're a Samoyed, is 15, which means you're 15 times more likely than a crossbreed to have diabetes. Now, that's fine and interesting if you're a diabetic researcher, but I think what's of more interest to this community is the right-hand side of this figure. Here is the boxer, the German Shepherd, and the Golden Retriever, and they have a very low risk of diabetes. In fact, we've been looking for diabetic boxers for my entire career. I've never seen one, ever. They just do not get diabetes. And actually, these are the dogs who get insulinoma. So this is where the diabetes insulinoma interest came from my perspective, because I think something about these breeds that's wired into the breed appears to make their beta cells very robust, but that might also predispose them to transformation to malignancy and immortalization. So that's where the Canine Diabetes Genetics Partnership was founded, to try to compare these two extremes. And just if you needed a little bit more convincing, I thought this was a very striking observation. Jerry Poulton produced a paper back in 2007, and he showed that there was a very unusual incidence of insulinoma diagnosis sort of peaking in the summer. And, you know, that might be some other thing that we don't really understand, or it might be simply to do with when dogs get referred, or dogs being outside, having more exercise in the summer, and that it manifesting more obviously. But actually, what's really funny is the way in which that mirrors inversely the relationship between diabetes and month of onset in canine patients. So we see a winter peak of diabetes and a summer peak of insulinoma. So the Canine Diabetes Genetics Partnership we founded to discover and understand genetic variants contributing to canine pancreatic beta cell health and disease. So not really just about diabetes, but also a really strong interest in insulinoma as well. And there are a lot of people involved here. I've listed them on this slide. There are many sources of collaboration and funding. A lot of our funding came from Pet Plan Charitable Trust. I'd just really like to draw your attention to Marsha Wallace here, or second from the right on the picture, who's done a lot of the bioinformatics of the project. And Brian Catchpole also, who set up the database and the archive of samples that we rely on so heavily. Now, you might wonder how easy it is to work on canine genome, because we don't have all the resources that are available for human genetics and human cancer researchers. We don't have the 100,000 Genomes Project. We don't have all the annotated ENCODE project and all the things online that we can use to look up all sorts of things like enhancers and promoters and transcription factor binding sites. So we have to be a little bit more creative, and it makes us also very collaborative. Now, the first canine genome didn't get published until 2005, and it cost a lot of money. And actually, I always like to show this slide in talks, because it illustrates here the article, the copy of Nature in which the canine genome was published. And here you have some dogs looking at Crick and Watson and the double helix, but you can see they have actually cleverly photoshopped in a dog to this picture. This is Tasha, who is actually, as it turns out, a boxer whose sequence is the canine reference, or was up until probably a year ago, and it's now, we have a Labrador and a German Shepherd as well. Both breeds of very good relevance to us in studying insulinoma. But this canine genome really transformed the way that we can look at genetic diseases in dogs, or also the transcriptome of tumours. And we can now do whole genome on as little as 200 microliters of blood for less than $800 a sample. The big challenge is working out, obviously, which of the genetic variants are most important, because if you look at what's unique to a boxer, some of that is going to explain why the boxer has a flat face and various other things, and some of that is also going to be about their predispositions to cancer. But there's a huge market for this. You know, people are really keen to know about their pets, and we have quite a lot of momentum from veterinary, pharmaceutical, nutrition industries and pet care industries to drive us towards understanding the molecular basis of breed-associated diseases like diabetes and insulinoma, so that people can make more precise decisions and choices about screening or healthcare, or which breeds they're going to have. So very briefly, I'm just going to outline what we've done in the Canine Diabetes Genetics Partnership from the point of view of the whole genome sequencing. And then for the final part of the talk, I'm going to talk about our work with insulinoma tissue. So this is looking at the genetics of the dogs who have insulinoma and diabetes. This is looking at whole genome sequencing. And in the first phase of this, what we did was we just took 32 of our patients, some of which had diabetes, some of which had insulinoma, some of which had neither of those things and had lived to an old age with neither condition. And this study, of the 32 dogs, 6 of those dogs were boxers with insulinoma, and 3 of those were Labrador retrievers with insulinoma. So that gave us a really good insight into whether there were any shared variants across those individuals. We then narrowed those down to what we call a twist list, so a much smaller number of genetic variants for a second round of study. We added some more insulinomas, some more diabetic and control dogs. And then we did a third round with another 120 dogs to narrow down those variants even further. And what we're currently doing is actually taking some of the genes that we found that are of interest and some of the variants, and we're looking at those in crossbreeds, and we're also now trying to understand what they do in cell culture with beta cells and with insulinoma cell lines to see whether they do influence the way in which the cells behave, whether that's to do with cell death and diabetes or cell viability, survival and growth in insulinoma. I'm going to show you a sort of summary slide of some of our results in terms of the boxes, but it's worth just flagging this as a particular feature of canine genetics research. When you're thinking about what variants contribute to susceptibility to a disease like insulinoma, we really divide those into 3 categories because of the way that dogs have a certain population architecture. We see outbred crossbreed dogs, but we also see within certain breeds really quite a small gene pool because they have been so tightly intercrossed to set a particular set of characteristics within that breed. Now, actually, that is in fact a natural experiment that helps us to try to filter down those genes and those variants that are important for breed-associated disease, but it does mean we have to think about things a little bit differently. So there are 3 different types of variants contributing to disease susceptibility. Let's imagine this is the boxer and insulinoma. There are variants which are high frequency and pretty much fixed within a breed. So these are present in the boxer, and pretty much all boxers have them. They might be exclusive to boxers, or they might be other breeds that get insulinoma as well. But all dogs carry that increased susceptibility, and then we think there'll be environmental triggers that might contribute to actually the development of the disease. So we call those class 1 variants. There are also variants purely within the boxers that you would see more commonly in dogs who have insulinoma than in dogs who don't have insulinoma. So that's almost like a case control study within a breed. And we expect some of the insulinoma variants actually to be like that. And then we've also got some very rare individuals who have certain gene mutations in oncogenes that might directly contribute to a cancer being caused in that individual animal. So we've also got to remember that association doesn't necessarily imply causation, but association is a really good place to start with the biology. And this is a summary really just to show you an example of what we've discovered. So this is class 1 variants. We do something called discordance analysis. We try and say what's so special about the Samoyed that makes it get loads of diabetes versus the boxer, which means it never gets diabetes but gets lots of insulinoma. And this is a summary of an awful lot of sequencing and bioinformatics, which I haven't really got time to go into. But what you can see here is a heat map. So if the allele frequency is near 1, that means that variant is carried in almost all of the dogs in that breed. And if it's down here on the dark color near black, the 0, that means it's never seen in that breed. So here are a collection of variants, our top hits, if you like, from this study, where we can see the Samoyeds are completely fixed for these genetic variants, and the boxers never have them. And then scattered through various other breeds like the Greyhound, the Schnauzers, the Cavalier King Charles Spaniel, and then a couple of large databases here at the bottom, the Dog Genome Project and another cohort from the Animal Health Trust. We can see that throughout the dog kingdom, they're kind of mixed about, but they've really segregated between the Samoyed and the boxer. And that's where we think our beta cell health and disease genes are. And you can see that we've color coded them along the bottom here. The colors show you whether these are known to be diabetes genes or immunity genes, metabolic genes, or purely pancreatic associated genes in terms of what those genes are actually encoding. So canine insulinoma can also be managed surgically as well as medically. Now, medical management is used when there is metastatic disease or excision has been incomplete, or the owner decides they don't want to proceed with surgery. And that's based on high protein, high complex carbohydrate food, short walks. The owner carries food or glucose syrup with them. We often give them corticosteroids to maintain their blood glucose. And there are other drugs like toceranib, receptor tyrosine kinase inhibitors, for example, that do seem to offer a survival benefit, although we're actually in the middle of a clinical trial at the moment to determine if that is just an observation that we think we're seeing or whether that really is happening, because that will also give us an indication as to whether that should be added to our armory of treatment. But what we see when we look at survival cohorts is actually we get much better survival with primary surgical treatment. If you do surgery and then you institute medical treatment, if the clinical signs relapse, obviously that improves survival even further. And this is a paper from 2007. I'll show you a 2020 paper that's got more dogs in it. But this paper is useful from Jerry Poulton because it does actually have a table in it that indicates the average survival time across multiple different studies. And you can see they're sort of between 18 and 47 dogs in each of these studies. And we're seeing survival times somewhere in the region of 1 to 2 years at most. Whereas this study here did show a much longer survival time with this medicine approach after surgery. So this is the most recent study just published in Journal of Small Animal Practice, taking cases up into 2020. And again, you can see on this Kaplan-Meier analysis that the surgical treatment dogs do better than the medical treatment dogs. And of course, we have to remember these dogs are diagnosed quite late in life and quite late in the disease course. So that's why I think we do see that many of them succumb not necessarily to the disease, but to other things as well during the 60 months following diagnosis. So we have talked a lot about how we might be able to improve targeted insulinoma treatment. Our concern as vets is that all we're really doing is managing the clinical signs, telling the owner how to deal with hypoglycemia, knowing all the time that that is getting worse. We do try to make the surgery as radical as possible. You know, we have to preserve some pancreas, but we also try to take as much of the tumour as possible. And if there's no obvious metastatic spread, then we can have some very good outcomes. I have even managed patients, unbelievably, who've had an insulinoma removed and have gone on to become diabetic. So we've had to give them insulin once the tumour has been removed, even though 50% of the pancreas has been preserved. So I've talked about the genetic basis of risk and the whole genome sequencing work that we're doing. But the other thing I just want to mention is the biological tumor behavior study that we've been interested in, and that's our single cell transcriptomics. Now, I'm sure many of you are already familiar with this and have seen this in other settings, but just for those of you who may not be so familiar, instead of taking the whole tumor and putting it into a solution to fix it and then extract the RNA and look at the average gene expression from all cells, a bit like one of the studies I showed you towards the beginning of this talk, single cell analysis allows us to look at each cell individually, and then we can look at each cell's distinct gene expression profile. We can plot them onto a plot like this, and we can look at small populations of cells and determine whether they are having an important role, their presence or absence is important in downstream survival or tumor behavior or response to treatment. So it has been said that single cell transcriptomics is a bit like taking a fruit salad, and instead of making a smoothie, what you do is you reassemble the fruit into its component parts, and you can look at it in more detail. Now, what I would say is that is true, but actually what you can't tell from single cell transcriptomics, which I think is a limitation, is you can't actually see the anatomy, because you have to break down the cell and the tumour into its constituent single cell parts to do this. So we're also really keen now to start looking at what's called spatial transcriptomics, to understand the anatomical locations within the tumour microenvironment, because whilst this is really helpful, I don't think it's going to give us the whole picture. So we use single cell RNA sequencing. We've already presented some of this data at one of the American College of Veterinary Internal Medicine forums, and it helps us to dissect what's going on pathologically, to look at the unique features of the malignant cells and study what's going on in the cells around the tumour. And we looked at 2 unrelated boxers diagnosed with insulinoma. I'm sorry about the bad photography there, because that was me taking that photo. That's why it's blurred. But this was the tumour that we used for the histological diagnosis and also for the single cell. And this dog actually did incredibly well. The dog from whom this tumour was removed and needed no treatment for a long time, and actually succumbed in old age to something else subsequently. So this first dog that we did actually was a relatively newly diagnosed and not hugely malignant tumour. The second patient had a more severe presentation and was seizuring prior to surgery, and that also matched with the fact that we saw a metastatic lesion in the abdomen that was also removed. So we took the tumours and we did single cell RNA sequencing around 3,500 cells in total, 2 tumours and 1 metastatic lesion, and you can stain them here to show that you have actually got beta cells. For those of you who are interested, we used the 10x Chromium system. We aimed for around 1,000 cells per tumour, and we sequenced on a HiSeq 4000, and we have a bespoke bioinformatics pipeline, which I'm happy to talk about if anybody wants to discuss. Again, for those of you who are interested, we use 75 base pair paired end sequencing. We got a really good number of reads per cell, and we found expression of around 1,000 genes per cell that we were able to detect. And you can visualize the data in lots of different ways. I'm going to show you a Tisney plots, but we typically now, in the new generation of sequencing, will use UMAP plots. And what we saw were really distinct cell clusters by their transcriptomic profile, and they were largely represented in both tumours. So a Tisney plot looks something like this. A UMAP is similar, slightly different way of performing that dimensionality reduction, but every dot on this plot is an individual cell, and the cells are clustered together by how similar they are in terms of their transcriptomic profile, and they're coloured by cluster. The software attributes cells into different clusters according to their profile. So this is a really basic summary of quite a lot of work and quite an expensive experiment, because single cell sequencing sadly at the moment is quite costly. So you can see here, this is the first case, and this is a tumour, and this is the second case that's a tumour, and you can see that actually there are some similar clusters in both cases. You've probably, your eyes been drawn to this orange one here, which is present in case 2, but not case 1. And actually, if you look at the metastatic lesion from case 2, it's almost identical to the primary tumour. But what you really need to try and understand is which cells are in each cluster. And the biggest surprise for us here was when we coloured those clusters according to the gene expression, and we asked which cluster is producing insulin, expecting it to be 1 and thinking there'd be 1 insulin secreting cluster identified. There were 2. So if we go back here, this orange cluster and this red cluster were producing insulin. So much less apparent in the tumour that did very, very well post-operatively, much more apparent in the dog who was seizuring. And if we look and attribute gene expression profiles to the different clusters, we could use data from other experiments to work out which were the exocrine cells, which were the immune cells, which were the insulin secreting cells, and so on. And it was really unexpected to see these 2 clusters expressing insulin, and also to see how they were different between the 2 patients, and to see how similar the tumour and the metastatic lesion were, suggesting that metastatic lesion was carrying with it not only tumour cells, but also cells that make up the microenvironment around the tumour. So when we look at an insulinoma cell, when we look at the single cell profile, we see that actually there are two parts of this pie chart effectively that are insulin positive. Everything else doesn't express insulin. We see a lot of endothelial cells, so vascularization is clearly important, quite a lot of immune cells, and two sorts of stellate cells, non-immune cells in that profile as well. And we can use heat maps to look at the different genes expressed in each of those 10 clusters that we managed to divide the cells up into. And we can see that orange one and the red one here. We can see their top 3 gene expression profiles. So this is the orange, and you can see the orange cluster's top 3 genes are insulin, chromogranin B, and islet amyloid polypeptide. Whereas if we look in the red cluster, which is here, actually those are not the top 3 genes in that cluster. In fact, insulin does not make it into the top 3 expression genes that are expressed in that cluster. So there clearly are 2 distinct clusters here. And this just, again, tells you the different properties that we attributed to each of those clusters. So, as I've mentioned already, one of the clusters was defined by high expression of the chromogranin B and islet amyloid polypeptide, and the second was defined by high expression of chromogranin A, MAP2, which is another microtubule-associated protein, and secretogranin. And the thing that really struck us here is that chromogranin A and islet amyloid polypeptide, so in both one in each cluster actually, have both been studied as diagnostic and prognostic markers in endocrine tumours. I'm sure many of you are very well familiar with that, but we thought it was striking that there were 2 different markers there in 2 different clusters. So we may be having to look at more than one cell type to make prognostic indications. So what does this mean? Just to wrap this up, well, it means we need to study more tumours so that to see if we can predict outcome from this type of data, to see whether any of those differentially expressed genes could be treatment targets. And we're also interested in using some of this data, maybe with bigger than 75 base pair sequencing reads, to see if we can find any mutations between the tumour and the germline genome. So I think we've demonstrated that whole genome sequencing has demonstrated several genetic variants which may predispose boxers to tumours. And we've got the Vet Compass manuscript to describe insulinoma epidemiology coming through. And I think we've demonstrated that single cell sequencing is feasible in dogs in fresh neuroendocrine biopsy tissues. And I think it has got really exciting potential to find new treatment targets. Our limitations are the cost of the technique, and also you have to do this fresh. We are working with insulinoma cell lines to try and get a freezing protocol so that we could batch these and do them more cost effectively. And our future plans really are to look at more insulinoma samples from more breeds, to look at for mutations. We're doing the Toceranib clinical trial being led by Florin, and we are also working with a range of cell lines from different species so that we have an in vitro way to try and model some of these genetic mutations or changes in transcriptome and see what that does to the survival of the cells. And I think I mentioned when I was introducing single cell transcriptomics, we'd also really like to be able to do this in spatial transcriptomics too. We'd like to be able to look at the anatomical relationships between the different insulin secreting cells in the insulinoma and determine whether that gives us any insight into which cells are going to metastasize and which are going to survive and which might be targetable through medical treatment. And ultimately, what we like to do is try to bring together the relationship between insulin and insulinoma and diabetes to try to understand more about normal beta cell biology too, because that helps us to recognize what's abnormal in neuroendocrine tumors. So I hope that's given you a bit of a flavour of what we do. I'd like to thank once again my colleagues and collaborators. I'd also like to thank MRC for my funding and various other charities who've given us money too, particularly PetPlan for the canine diabetes work. And I've put my email there at the bottom, ldavison@rvc.ac.uk, if anyone wants to get in touch. but I'm also happy to answer any questions as well if people would like to ask about canine insulinoma. Thank you very much for your attention.
Thank you so much, Lucy. I have to say, I find all of this completely fascinating, both as a proud dog owner and as a researcher looking at insulinomas. And I do think that this has sort of been an underutilized resource for us, actually, in the sort of human research field, that there seems to be some striking similarities in the canine insulinomas compared to the humans, both in genetics and treatment and sort of phenotypes. Has anybody got any questions before I carry on chatting?
So I just wanted to say thank you. That was an absolutely phenomenal talk. You did a great job of, going into the science, but also keeping it simple. And I could follow along. And I just want to say thank you. But the other thing I wanted to bring up, I was very excited about this talk because so I'm a basic scientist that studies human neuroendocrine cancer. And I do mouse studies, and I work very close with the lead vet here at my institution. And he helps me with the procedures and whatnot. And one day we were talking about what I study, and I told him that I was studying, insulinomas and neuroendocrine tumors. And he said, wow, I see those all the time. He said when he was working at not an institution, but when he was working at like a private practice vet, he said he sees insulinomas all the time in ferrets. And he was like, why are you studying in mice? You should be studying in ferrets.
Absolutely.
I said, I had no idea.
Yeah. I didn't put that in the talk, but ferret, you think dogs
You've
get
seen that
insulinoma?
too.
Oh, ferret endocrinology is crazy. Adrenal tumors, insulinomas, they have a very strange also, females have a very strange bone marrow toxicity they get if they're not mated. There's really, really fascinating ferret endocrine disruption going on, and whether that's what we don't have are people who own ferrets who have that sort of relationship with them that they really want to
That's what he
them.
said. It's, he don't do surgery on a,
but
ferret.
actually part of our remit in terms of what we're trying to do with our multi-species functional genomics lab and the spatial transcriptomics is that if we can establish a protocol for archiving those tissues, even post-mortem, and then we can come in and do spatial transcriptomics or even just do single nuclear preps and single cell, then I think that will reveal a lot of really good information. But yeah, the whole world of ferret endocrinology, I mean, you think canine and feline endocrinology hasn't had much attention. Ferrets, we all know they're there and we all know they get insulinoma, but actually typically people just go, oh yeah, it's got that ferret low blood glucose thing, we'll get another ferret, sadly. That's just but yeah, you're exactly right, and it's the case in the UK too, definitely.
Wow. Yeah, I recently learned that. So there's actually my institution has a large ferret research program because they use ferrets to study cystic fibrosis.
Right. Yeah.
And so the ferret room's just down the hall. And so I wonder if it's worth my time to ask them.
I would. It depends how old they are, because they have to be a certain age to develop those tumors, but absolutely, and anyone who's got an interest in adrenal tumors as well would also, find ferrets interesting.
Yeah, I have an endocrine surgeon on board. She really wants to study the ferrets. So maybe, although they're used for cystic fibrosis studies, I thought to ask them, if some of your ferrets develop insulinomas, I would love to
definitely get tissue.
Look at that.
What we're also learning is that we can go back to archive tissues and do more with it than we ever used to be able to do. So, just even getting to put them in RNA later and freeze some tissue, then before you know it, you'll have a bank of tissue, and we can talk again about where we've got to with the dogs. Yeah, absolutely.
So yeah, if I get my hands on some tissue, you'll get an email from me. So
definitely, that sounds great.
Yes, thank you again for a wonderful talk.
Perfect. Anyone else got any questions? We've got like 5 minutes or so. I have many questions that I can ask, but otherwise no, yeah, I find this. Oh yeah, go on.
Oh, sorry. Yeah, I thought that was a really good talk as well. I think that you're really approaching this pretty well. I'm also a neuroendocrine tumor basic scientist for human research, and one of the things that we've kind of noticed over the years, there's not a lot of targetable mutations in terms of the pancreatic neuroendocrine tumors. You really don't see it a lot in any of the big genomic studies that we performed. So it's really cool seeing you take this on, but I'm wondering, I've got, random questions all over my head, but about like how kind of it works for dogs. But my kind of take on that, I think I'd be really interested to see, because you have these different dog breeds that are getting at different rates, if there's some sort of, I don't know if it would be more genetic, but probably more either like tumor microenvironment related things, like the immune cells themselves having differences in their ability to recognize these kind of tumors. That's something I think would be really cool to look into.
Absolutely. Yeah, I mean, obviously we're interested in the MHC because that's a big thing in type 1 diabetes. It's not really clear whether dogs get type 1 diabetes. You'll read that in the vet
That was one of my
textbook,
other questions. Yeah.
But it probably isn't that, to be honest. It's probably a slightly weird, like latent autoimmune in some dogs. In some dogs, it's pancreatitis. In other dogs, it seems to be insulin resistance. And actually now we're starting to see other monogenic forms of diabetes that we didn't know, but they were monogenic in certain breeds. So yeah, I think you're right. And it's remarkable the inbreeding that's happened in dogs. There are some dog breeds who've only got 2 MHC haplotypes. So you think actually this could actually have a huge amount to do with the MHC. And it's only really the class 2 MHC that's massively been studied in dogs. Class 1 isn't particularly well studied. So again, that will be another area that I think we should be addressing, because we definitely see MHC class 2 expression in these tumors. So yeah, you're right. That will be, that kind of immunological microenvironment is definitely, and that's why the anatomy becomes so important, doesn't it? Because we sort of need to know what's next to the tumor as to how it's actually being treated by the body.
Yeah, and then the fun kind of weird thing I was thinking, you showed all the breeds that are kind of on the spectrum there. With modern era, dog selective breeding, there's a lot of different crossbreeds now. So I'm wondering if you're going to start seeing some of these very, high insulinoma being crossbred with very low insulinoma rates, and seeing, do they lean more towards the diabetes side, or do they lean towards the insulinoma? I think that would be really fascinating.
I think it is. I mean, I'm sure you have it in the US. We have a massive explosion of oodles at the moment, everything oodle. And our medical wards are full of oodles. It seems not to be that particularly doesn't a lot of, whether it's just there are a lot of oodles out there, or whether there is something bringing together things that are somehow permissive or things that are high risk, and poodles themselves or miniature poodles at least are a high risk breed for diabetes. So you wonder what that's doing to the mix. And then also in the whole world of the oodles, as well as having the 2 primary breeds being crossed, there's a definite new layer of intercrossing the F1 oodles, and we're seeing even more problems coming from that. So I think where you have selective breeding and a minute, as reducing gene pool, it does, it causes welfare issues that we really try very hard to educate people about. But actually, scientifically, it leaves us with, sometimes some of the selective breeding experiments do leave us with a much easier job of trying to find those, important variants. And you're right, the next generation coming through, well, you'll see in the vet, just muted myself, the next generation coming through, it'll look different in 10 years time, because everyone's buying French bulldogs and oodles and pugs at the moment, whereas a few years ago it was Labradors. So we're going to get these waves of different things coming through.
Thank you.
Thank you. This is Sonia. I'm actually a patient, and I was diagnosed back in 2002 with a carcinoid tumor that then reverted to, after a resection, reverted to Cushing's syndrome. And then when I started to give talks, and I'm lying there in the hospital bed, and I'm going, how come dogs and horses get this? And why did I get it as a human? So for me, it was to turn it around psychologically and be there for other patients and be there to teach doctors. I have found your presentation today so interesting, because I found also that these animals were diagnosed much sooner than a human patient. And that's a crying shame, in the sense that patients can take up to 15 years for a diagnosis, whereas
I've read that. Yeah.
it's much earlier.
I think, yes, I think that is true in terms of the ones that we are recognizing. They often do come to us quite quickly, especially if they're insured. I wonder if we are missing a proportion that aren't being ascertained because they're just not coming to us. I think that would be the case. But I think in veterinary medicine, because effectively it's private healthcare and there are sort of subspecialists within it, then and insulinoma is a bit more common in dogs, I think, than humans. Perhaps gives us a better shot at it. But that's really fascinating. And Cushing's syndrome, we see in dogs and we see in horses. And the thing about the horses, actually, and I'm aware we're approaching 7 o'clock, but the really interesting thing about horses is they get Cushing's syndrome, they get hypercortisolism, and they get really, really dramatic insulin resistance, massively high blood glucose, they get laminitis and all of these other signs. They never run out of insulin. They never become diabetic. Now, I'm not sure I would ever be in a position to know if a horse had an insulinoma because I'm not an equine vet for a start. And I think, if there was ever any sign of a horse becoming wobbly or collapsing, nobody would necessarily pursue that. But it is incredible that the pancreatic beta cells of the horse are somehow still able to make all that insulin without becoming neoplastic, whereas in the boxer, that seems to be at the expense of the syndrome. But the Cushing's in dogs typically is driven by the pituitary, making the adrenals produce too much cortisol. But we do see primary adrenal hypercortisolism as well in dogs and sometimes in cats. And cats can get insulinomas as well, but it's pretty rare.
Yeah. Thank you so much.
Oh, thank you.
Well, thank you. I mean, it's now 7 o'clock. I have to say that I could chat all day. I've got many questions because my expertise is epigenetics. But I think I'll be in touch definitely with you in the future.
Always happy to talk about dogs, diabetes, pancreas, tissue, insulinoma. Very happy, very willing to engage. So yes, please just drop me an email.
Well, yeah. Well, thank you so much for coming and giving this talk. And I always like research that's a bit outside the box from the human stuff. And I think this has been fascinating. And I'm hoping that it's the start of a lot of conversations that people will be having to think about this sort of thing in future. And I'm sure that it will do nothing but benefit the patients because, as I said already, it's ridiculous that dogs can be diagnosed quicker than people, which is not an ideal.
It sounds like it's a very stressful journey for those who have to go along it, and anything that we can do that contributes even to shortening that journey is helpful, I think, because that's a hugely stressful part of the process, I'm sure. Thank you very much for the invitation, and hopefully I will speak to some of you at some point again soon. Thank you.
Yep.
Thank you.
I just
Thank
you. Thank you very much, Lucy. That was absolutely fascinating. I mean, one of the most interesting ones and one that I can follow because I have no scientific knowledge at all. So fantastic. And thank you everyone for coming. Just a reminder, the next meeting is on the 25th of April. So hopefully we'll see you all then. But thank you very much. Thanks again, Lucy. Great. And thanks, Kate.
Thank you.
Thank you. Bye.